everyone. Thank you for joining us today for the pre-application webinar on NCI's Provocative Questions, Funding Opportunities Relevant to Population Scientists. Before beginning our presentation, I would like to provide a few tips for using WebEx so that you get the most out of today's webinar. All attendees will be in listen-only mode. Please feel free to submit your questions throughout the presentation in the Q&A or chat panel and select all panelists from the drop-down. A moderator will ask the questions on your behalf during the Q&A portion of the webinar. If you need to view live closed captioning, please refer to the media viewer panel. I did want to note that today's webinar is being recorded, and with that, I will pass the presentation over to our presenters.
Hi, this is Stephanie Nelson. I'm going to start us off for today, and first I'd like to introduce our other presenters, Kelly Filipsky, Danielle Carrick, and Jill Reedy, and we're all program directors in NCI's Epidemiology and Genomics Research Program. So first, our agenda, we'll start off with a little bit of background on the Provocative Questions Initiative. We'll talk about some specific opportunities for population scientists. Then we'll give you some information about the application process, let you know about some resources related to the PQs, and then at the end we'll have time for questions. So, the NCI's Provocative Questions program is housed in the Center for Strategic Scientific Initiatives. It was designed to stimulate specific areas of cancer research that were seen to be understudied, neglected, paradoxical, or that have been difficult to address. The PQ program was established in 2011, and it was designed to support projects that would solve specific problems in cancer research that had been identified by the NCI as a provocative question. The PQs don't represent the full range of NCI's cancer research priorities. Their purpose instead is to challenge cancer researchers to think about specific problems that are important but that may not have received sufficient attention. Since 2011, the initiative has received more than 2,500 new R01 and R21 applications and has funded over 230 of these applications. So the PQ questions are developed by NCI staff, and to identify and select questions, NCI sponsors several workshops with experts in the extramural communities, and they get together to identify and prioritize compelling but understudied problems in cancer research. All the questions are reviewed by NCI program staff from all divisions and are submitted to NCI's scientific program leadership for final decision. So our process for soliciting and choosing PQs has changed somewhat over the past few years, and when the next time comes to develop new questions, we'll try to make sure that the epidemiology and population science community is notified and can have some input. A couple of us here served on the group that reviewed the proposed PQs last time around, and we found that there is interest in soliciting research ideas that align with population health priorities. So the scientific program contact for this program is Sean Hanlon, and we have his email listed here. After the PQs are selected, each question is assigned a team leader and a team, and these teams include 4 to 8 program staff from all NCI divisions and centers. So in the next slide, I'm going to show you a team roster, all the members of each PQ team, and you'll see one of the rows highlighted in blue, and that's members of the Division of Cancer Control and Population Sciences who serve on the different question teams. So next we're going to highlight some questions that are led by or on which DCC PF staff are involved and that we think might be of interest to people in the population sciences field. So, So I've listed 5 PQs that we think might be of particular interest to population scientists. The first, PQ1, asks how molecular mechanisms might influence disease penetrance in individuals who inherit a cancer susceptibility gene. PQ3 looks at genetic interactions between germline variants and somatic mutations and how these might contribute to differences in tumor evolution or response to therapy. PQ6 looks at circadian processes and how these might affect tumor development, progression, and response to therapy. PQ11 is a question asking whether diet and nutritional interventions affect response to cancer treatment. And PQ12 focuses on molecular and cellular mechanisms that might underlie the development of cancer therapy-induced adverse events. The next few slides are going to show each PQ and a paragraph about each, which is the intent of the question. And one thing to keep in mind is that the PQ intent is a little broader than the actual question itself might seem. So keep this in mind when you're thinking about whether you have a research idea that conforms to one of the questions. And we'll discuss responsiveness to questions a little more in our section about review. So the first question, what molecular mechanisms influence disease penetrance in individuals who inherit a cancer susceptibility gene? And the goal of this question is really to go beyond association studies and really try to determine why certain individuals who have a known cancer susceptibility gene, here we have some examples, Li-Fraumeni or Lynch syndrome, why not all individuals with these mutations develop cancer, or why some have increased risk versus others. I think the main point of this one is to, when I think of population health scientists, is to think about going beyond your association data and sort of understand more what's going on beneath this association. We have funded a couple of DCC PF investigators through this question, and one study looked to identify genetic differences between twin pairs, discordant and concordant for breast cancer, and also determine how differences in their behaviors, environmental exposures, and certain developmental differences might affect breast cancer occurrence in these women. And we have a second investigator who was funded who was looking at the role of CDK2A germline mutants in families with pancreatic cancer and melanoma. This group was looking to identify candidate modifier genes and also create a model that would incorporate genetic, biological, and epidemiologic data and determine how these affect development of these cancers. So now I'm going to turn it over to Danielle Carrick, who will tell us about PQ number 3.
Okay, so PQ number 3, this is a question on do genetic interactions between germline variations and somatic mutations contribute to differences in tumor evolution or response to therapy. Now this was a question that was new for the July 2017 receipt date, so we've had 2 receipt dates for it. And we have received between about a third and about a half have been population science studies. So the intent for this one is that it's looking, we know that cancer, is characterized by numerous genetic and epigenetic alterations occurring in both the germline and somatic genome. So independently, cancer projects focused on characterizing the germline and tumor genomes have really led to improved understanding of cancer etiology and biology. So this PQ expands on that substantial investment that NCI, as well as many other places, have made in characterizing both the host and tumor genomes, and it asks for experimental approaches that integrate germline and somatic genetic data to better understand cancer risk and etiology. The main thing that we've been observing when letters of intent come in is that if there's an issue with responsiveness, it's generally because the proposal is not fully integrating, not really integrating germline and somatic genome. Okay.
So provocative question 6 is looking at how do circadian processes affect tumor development, progression, and response to therapy. This was also a new question for the new RFA, so we have had 2 receipt dates. And the purpose of this question is really to understand how circadian and other oscillatory processes influence cancer development, progression, and response to therapy, and how that can illuminate potential new avenues to manage or treat cancer. So far, they've received a lot of basic science applications for this question, but this is a good question to to think beyond the basic science and look at this in epidemiologic studies. And unfortunately, our team leaders couldn't be here for this one, but if you have questions, please contact either Sean Hanlon or Lisa Galicchio here in DCC PF.
For PQ3, we're really looking for applications that address how diet and nutrition interventions affect the response to cancer treatment, treatment-related adverse effects, cancer prognosis, and also related health outcomes. So here we're encouraging all types of studies that can help to elucidate the mechanisms for how diet and nutrition affect cancer outcomes. And this includes clinical trials, observational studies, and mechanistic studies in both animals or humans. Again, we're looking for transdisciplinary research and studies which might consider things like the multidimensional or dynamic dietary patterns, where we can think of dynamism as including timing of meals or circadian rhythms, as well as changing diets over the course of treatment and survival, and that those would also be encouraged as well.
And provocative question 12 is looking at what the molecular and cellular mechanisms that underlie the development of cancer therapy-induced severe adverse sequelae. And this one is really looking for research that identifies novel mechanisms that induce chronic sequelae, apply that knowledge gained to facilitate and design new treatments or approaches that may decrease or reverse these adverse events, and facilitate mechanism-based design of new cancer therapies that are expected to show decreased adverse effects when compared with existing therapies. Now, this question appears that it's very mechanistically focused. However, we are encouraging population scientists to also bring applications into this one. It's not required that you have a mechanistic aim in your grant. The question just really needs to be based off of a mechanistic hypothesis. We do accept preclinical, non-clinical, and clinical settings for this. And we have unfortunately not received any applications that were population science yet. But we would really like to see some that are maybe translating some of the mechanistic findings into other avenues.
Okay, I realize we went through some of that a little quickly, but all this information on the different PQs is available at the Provocative Questions website. And now I'm just going to quickly show a couple of slides that show the staff members for these PQs. Again, if you want to contact a specific person, you can go through Sean Hanlon. His contact information is on the PQ website. Now I'm going to turn it over to Kelly, who's going to talk a bit about the application process and a little bit about review.
So for the application process, the current RFA has 2 receipt dates left for June 28th and October 30th, 2018. For this FOA, letters of intent are recommended but not required, but I want to emphasize that this is a really important step in this process. At this point, you're able to send in a letter of intent, and if you include your aims and a little bit about your project, it's sent to the PQ teams, and the PQ teams are able to look at it and let you know whether or not they deem it responsive. And at that time, you'd be able to talk to a PD from one of those teams that can either direct you to a different mechanism if it's not responsive, or try to help you fit it in to the question so that it does become responsive. So this is an opportunity for you to make sure that it's not going to get rejected when it is submitted as a PQ, and it's a great opportunity to reach out to the PDs and really be able to speak with them. And all of this would go through Sean Hanlon. It's usually done very quickly, and we'll get to talk to someone from the team within a week or two from sending in your LOI, if not quicker. So I really highly recommend and encourage you to do this, and you can reach out as early as you want, and we're happy to talk with you at any time. And again, these all go through Sean Hanlon, although if you want to reach out to us specifically, that's fine. Now, once the applications are submitted, they are grouped together and sent to the PQ teams. And at this point, the PQ teams go through each application, and we compare it with the intent statement for each question. And this is how we deem them responsive or not. So that's why we encourage you to really take a look at those intent statements and see, some of the broad applicability that they have, because that's what we're looking at to really deem these responsive. And we have here a link where you can go and look at some of the PQ grants that have been funded through this RFA. So for the FOA, we have several different funding mechanisms. There is an R01 and R21, and then we have revision applications for R01s, U01s, P01s, and P50s. For the R01 applications, there is not an ARA process at the moment, so there aren't budget limits. However, we do look at the budgets very heavily like we do most grants. These are usually 5-year budget periods for these grants. Our R21s are the small grants that are really meant to be exploratory in nature. Your direct costs are limited to $275,000 over a 2-year period, and they can't exceed $200,000 in any single year, and your budget period cannot exceed 2 years. This mechanism for the R21s is a really great time if you have an R21 application to get it in. This is a great mechanism for that, because the purpose of the provocative questions is that they're these exploratory, high-risk, high-reward, and that's sort of what the R21s are as well. Now, the revision R01s, these are essentially a supplement to an existing NIH grant. So that can either be an R01, U01, P01, or P50 that has been funded through any mechanism at the NCI. It does not have to be a PQ grant. Your question that you're addressing in the revision just has to fit into one of the provocative questions. And for these, the direct costs are limited to $150,000 per year, and you can request support for up to 2 years. So these are a really good mechanism if you have one aim or 2 aims that can expand the scope of your currently funded NIH grant, and it fits into one of these questions. It's a great way to get a little bit more research out of that, and we're happy to answer any questions about these mechanisms. And I just want to mention that each of these has its own FOA, so you will have to go through whatever FOA that is when you're submitting. And we do allow resubmissions as well. So if you have an R01 and it came back and it didn't get funded, within the next 2 receipt dates, we can accept resubmissions for this current FOA. So the PQ since 2015 have funded quite a few applications. These are the R01, R21s, and our revision applications that have been funded. In parentheses is the number of DCC PF grants that have been funded. And this is a number we'd really like to see increased. This isn't because we're getting a ton of applications and they're just not getting funding. We're not getting the applications. So we'd love to see a lot more population science get sent into these PQs. And so these are the links for the current PQ RFAs. Now, I will say that when you click on this link, it will tell you that the RFA has expired. It has not expired. This is due to the clinical trial changes that are happening at NIH. So what's going to happen is that there will be a reissuance of each one of these RFAs. They may have a different RFA number, but they're going to be clinical trial optional to fit into the new clinical trial regulations that we have here. So everything else on that RFA will not change. It should be reissued shortly. And we do still have receipt dates that will be in June and October. So that may look very confusing, but we can answer any questions that you might have. And this should be reissued very quickly. So that should be fixed pretty soon. So at this point, we're happy to take any questions that you may have. You can type them into the chat box, and we can answer anything that you have.
So, while we're waiting to see if some questions come in, we have a few questions that we found people ask more and more frequently. The first one, how do I know if my idea will be responsive? As Kelly described, working with the PQ team is really a great opportunity for these questions. You can send your ideas into Sean, and he will distribute them to the PQ team, and we can discuss with you, if your idea is responsive to that PQ, to another PQ, or if it's close to being responsive, we can work with you to figure out how to make it more responsive. As Kelly mentioned, you can reapply if your grant is not funded. I think we went through revision applications, which are essentially supplements. How are the applications scored? The PQ applications are all given a priority score, and then applications to all the PQs are ranked. So each PQ is not ranked separately. If the PQs 1, 2, and 3 have all the top applications, those are the ones that will get funded. There's not really a defined pay line. It's really determined by the scores each round and by the budget that we have.
So to answer some of the questions we've received, the first is, will each PQ have its own study section? So these are broken into thematic groups. I believe there are 4 now. So there are, I think, 3 questions per group. So there is some overlap in terms of expertise needed to review these applications. So they all get reviewed into these smaller, very specific study sections, which we found has been really great reviewing the science. So the next one is, may we get the list of contact people for each PQ? This is being recorded, so you can look at those lists. You can also contact one of us if you have a specific person in mind, or Sean Hanlon can direct you to any of the right people to talk to. He usually just sends to the team lead, and we distribute it to the right expertise. So feel free to reach out to any of us. Our email addresses we'll put up here in a minute. What do you consider PQ2 and PQ10 to be not applicable to population sciences? So it's not that we don't consider them to be applicable to population sciences. All of them could be. These are just some of the ones that we aren't seeing a lot of population science applications coming in. So we want to encourage population scientists to do that. I've talked to a lot of PIs that when they look at the PQs, they look very mechanistically focused as opposed to population science. And so we wanted to kind of dispel some of that myth and really reach out to the PIs we work with and encourage them to submit to this RFA, because it's a really great opportunity for funding. Next one is for PQ10, which was not discussed. One question I have is how cancer therapy defined. Is this pharmacologic, or can it be surgery, for example? I'm not specifically sure about that, but I think it could probably be both. That would be a good question to reach out to Sean for, and he'll send you to the right person for that question. How important are preliminary data for R21s? So R21s are exploratory grants. So these do not have to have the amount of preliminary data that an R01 has. They will be reviewed with other R21s, so they'll have a similar amount of preliminary data. And again, these are really set up to be exploratory small grants. So the level of evidence necessary for these is a lot lower than an R01. But they do kind of look at whether or not these are innovative and very exploratory new ideas for an R21. So the next question is, how do you deal with overlap between PQs? For example, the microbiome in therapy shows up in both PQ10 and PQ11. Jill and I have actually dealt with this a couple of times because PQ12 and PQ11 tend to overlap a little bit. And what happens is we discuss it amongst our team and with Sean and decide which one it fits better in. We kind of know what the makeup of the study section is, so we can kind of tell which one it should really be reviewed in. So, we can handle that. You're welcome to speak with us, and we can talk about it and let you know which one we think would be a better one. And Sean, he listens to every single review and knows everything that's going on, so he is a great resource. And if he doesn't have the right answer, he'll ask one of us.
Oh, I just want to also add that that's another good reason to submit a letter of intent, because once you submit your application, you have to put down what provocative question you're answering. Otherwise, it's not going to come through. So you need to make sure that you get that situated before you write your grant.
Mm-hmm. And one more plug for study sections. When you submit your application on your cover page, there is a space where you can put the correct expertise that you want on the study section. I highly, highly encourage applicants to put at least one or two expertise that they think is important for their application in that box, so that we make sure that the right people are on the study sections to be reviewing your grant. So the next question is, are the supplements reviewed by a STEP or a study section or by program? So these are reviewed by the same study sections that the R1 and R21 PQ applications go. So they will be reviewed by a study section with all of the other applications for those questions. Our next question is, are the rosters for the PQ study sections available like for other study sections?
I think they were on the website. I'm not sure. They should be available.
Yeah, they should be available if you check the website. If not, contact one of us and we can find that out for you. The next question is, are R21 and R1s reviewed separately? So if it's for the same question, they go to the same study section. A lot of times they'll be broken out within the study section of R01s and R21s. It kind of depends on the SRO that's running the study section. So they will be reviewed with other applications that have the same theme. The next question is, can you please explain the resubmission process since October 2018 is the last submission date? Sure. So that is the last opportunity to resubmit to these questions. If this RFA is reissued after 2018, some of these questions may stay, some of them may not. If it's the exact same question, then you'll be able to resubmit. If not, October 18th is your last resubmission date. So if you're submitting to the June application date, it would be a really, really tight turnaround to get it into the October one, but it could probably be done. So I would suggest if you're thinking about getting an application in, getting it into the first application date here is probably your best option. And the next, yeah, that does answer. Our next question was, is it possible to submit in June, get comments, and resubmit in October?
It would be very tight because I think the reviews usually happen in September, October. Yeah. So that would be very tight.
Yeah. Yeah, it'd be very, very tight.
Yeah, and if the PQ program persists, based on what happened last time, as Kelly said, a number of the questions probably will be retained. And last time, we had the October receipt date for the FOA, and we were able to get the new questions settled and published in time for the June receipt date again. So if things go as they have in the past, it should be pretty seamless receipt date-wise. You just have to take a look and make sure that the question you're interested in is still there. And also another thing is sometimes they will keep the question but change its number. This can cause a little bit of confusion. It's not career destroying though. If you put the wrong question number, we can usually figure out where it's meant to go. But just something to pay attention to.
So our next question is, when is the review date for June submission? That's usually in September or October. If it's in early September, you would at least have your scores for a resubmission. But if it's mid-October, you most likely wouldn't. So it kind of depends on when that's scheduled, but it's usually in September or early October. The next question is, what's the funding level? Is it 10%? So these have a set-aside fund, so there's a specific pot of money that this comes out of. And so each cycle, each receipt date, we base it on how many applications scored well. So that's kind of where we fit our funding level. So it changes from receipt date to receipt date. So we can't really say much about that. But they've been doing really well, is something we can say. So that's exciting. So our next question is, if you submit in October or not funded, could you resubmit under the parent R01? Yes, you can do that. It would just go into the parent R01 pool. The next question is, what determines what PQs are retained? Is it PQs that receive lots of applications or those that don't get enough? It's actually a little bit of both. We had some that were retained because of their popularity and some that were retained because they got very few applications. It kind of depends on what we see coming in, basically. If we see that there is a need, if it's a very broad question, it's gotten a lot of applications and we kept those. But we kind of look at what is the need in that specific area and what are the new questions that are being proposed. Some of them were changed slightly based on what was proposed in some of the meetings that we had. So they were same in terms of theme, but they were a little different questions. Or we might change it because first round was mechanistic and we want it to be translational the next time. So it just kind of depends on the applications that we're getting in and what the field looks like, and if it's, if we feel like we're significantly impacting the field.
Yeah, and it seems like the PQs have, in some ways, kind of jump-started some areas of research and given them a little bit of a boost so that now, we can consider other ways of funding this work. Is that it? Mm-hmm. Okay, I guess we have no more questions.
Great. So I want to thank all of our presenters today and our audience for such a great discussion and all of your questions that were coming in. If you have any questions that were not addressed during today's webinar, please feel free to reach out to one of the emails on the screen right now. I'll leave this up just so you can jot it down before we close out the webinar. I do want to say that there is a survey, and we would greatly appreciate your feedback when you exit today's webinar. And with that, thank you again for your participation.